Well, thank you for attending and thank you for the invitation. I'm glad that I can finally introduce our facility to a lot of chemists. I've never seen so many chemists in the attendance for kind of my work, which is what I'm going to show you is really like molecular and cellular biology facility and what we can do. I mean, I think our expertise is very complementary to what you guys have. So, just to introduce myself, I'm a professor at the INRS, Institut National de la Recherche Scientifique. For those of you who don't know where we are in Quebec. We are in Laval. It's a suburb of Montreal. And INRS is a network of university. It's four main research centers, 170 plus professors, all graduate students. We don't have any undergraduate students, so we steal the graduate students from other universities to get and attract them in our labs. Um, so what you see on the right is where we are. So we are on the campus of Laval, the building in brown. So we are on the campus, and this campus is really doing molecular and cellular, mostly research, with a big tradition of immunology and virology, and also any research that impacts human and animal health. So, the molecular and cellular biology facility that we have in our lab is vast. When molecular and cellular biology means is a pretty vast expertise we can offer to our customers. You have a list of the expertise we have and the services we provide and we can provide. In terms of cellular biology, we do a lot of cell-based assay development, optimization. Flow cytometric analysis is a bread and butter for us. We do a lot of flow cytometry analysis of cells and all sorts of cells. We can help researchers for cell binding studies, functional studies. We also carry out transcriptomic analysis, comparative studies between two cell types or two different conditions. And because we do transcriptomic analysis and other type of analysis, we do a lot of bioinformatic analysis in terms of treatment of data. That is also I'll show you a few examples of that. Um, so all sorts of cellular and molecular studies in terms of transcriptomics or mitochondrial or genomic studies, things like that. We do some productions of recombinant proteins, especially in bacterial expression system, using affinity chromatography mostly. And this is something I'm going to talk about at the end. Maybe something you are less familiar and that we've been using for many years is the generation and characterization of nanobodies. I'll explain what nanobodies are and how it could help your research. And of course, part of our services is the training for new users, whatever it's lab safety, best practice, use of the facility, and all the expertise we provide. Um, our main we don't have we're not a facility of with huge expansive equipment. Most of our expertise in service is in our brain, but we have some core facility. The main core facility where everything goes is done, studies are done, are in for the cellular biology is our mammalian cell culture facility where we have biosafety cabinets, as you see, several incubators and all the small equipment that you need to work on your cells. We have, of course, freezers, refrigerators. We have some freezers at 100 minus 150. This is an interesting feature, and this is like a gold mine. The number of cell lines that we have in our biobank is quite impressive. I would say hundreds, if not thousands, of cell lines for different kind of assay, mouse or human, or any animals actually. A lot of samples as well, but cell lines we have a lot. So often we are asked, do you have different kind of cell models? So we basically, most of the time we do. And that facility is, of course, run by an experienced research assistant, Marlène Fortier, who has been working on the tissue culture room with me for almost decades. Marlène is the boss of the cell culture facility. You know, even when I get into the cell culture facility, I ask Marlène if it's okay. So she's doing all the training, supervising, and everything. Uh, okay, and then I'll give you a few examples in the next few slides about what we are doing. One of the things we've been doing over the last few years, and that has been very useful, are multi-omic analysis. And when I say multi-omic analysis, I'll explain to you many examples of that. Our most recent one is the cell-based assays like the secretome, where we do high throughput analysis of about everything that is secreted by tissue by cells. So it could be when you're activating your cell models or you're developing inhibitors. In our case, we have a long history of that, of developing inhibitors of specific cell functions. We want to know what are the mechanisms involved. So we do these cell-based assay secretomes, which are very simple to carry out. They're not that expensive considering the amount of data you have. What we have developed really is on the right is really the bioinformatic analysis of all the data because you end up with data of hundreds and hundreds of factors, cytokines, enzymes, and the supernatants. It can be within the cells as well. And then we do bioinformatic analysis that will allow to tell you, give you a mechanistic image of your assay. Flow cytometry, we've been doing that for 30 years. Ask us, we'll do it, or we can do it, or we'll provide expertise on our experience of flow cytometric analysis. We don't have ourselves. We have actually two small flow cytometers in our lab, in our facility, but most of the time we go to our flow cytometric core facility at INRS, which has these big multi-parametric capacity for analysis. So what we do, we most of the time, and we offer our services for the experimental design. And this is very important for flow cytometric analysis. For those of you who have done that, I mean, all the variables that can impact on your results. We do a lot of panel designs in terms of if you're doing multi-labeling of cells. So I think the fluorophore selection, the antibody combinations, we can help with that. We do optimization of sample preparation protocols for specific cell types, whether it's adherent cells or cells growing in suspension. Controls, I've put it in bold because it's so important. I've seen so many people doing analysis, flow cytometric analysis without the proper controls. So we help on this. And we help for the validation of antibody reagents, instrument setup and calibration, data acquisition, manual gating. The gating is very important when you're doing multicolor analysis. And of course, all this is included in the training and technical support. Another thing that we're doing more and more in the facility are transcriptomic analysis, comparative studies. I'm giving you an example on the right where we have compared three conditions of cancer cells that were actually, this is a cancer cells that were the testing of inhibitors to see if the inhibitors we were developing can alter the secretion or the expression of mRNA genes in cancer cells. This work has been published. I haven't put the reference, but you can find it easily on the web. It was published recently. But you see, we can do it PCOA analysis, volcanoplots, and all these heat map to tell you that tells you what is the impact of your conditions. That we do a lot as well. Um, for glyconet, something that we have started to do a couple of years ago, and that was this is an example of an analysis we've done, we've published last year. This is looking at the glycogen signatures. We've got some at Glyconet in the past, a few talk about the glycogen signatures. It's an important concept in terms of glycosylation that is really emerging. It's working pretty well into humans. I mean, we have great databases for analyzing the expression of glycogens in human cells. Uh, it's getting more complicated when you get into different animals. In this one here, this is done in blue mussels in the national park in Park Canada. And that is demanding in terms of because the databases of glycogens for animals is not well developed at all. So we have to work it out. But it's getting there. I think we're developing more database, but we certainly can provide if you're looking interested into the glycogen signatures as biomarkers or mechanistic studies. We certainly can help you out there. We're doing it right now into a shark, a kind of shark that is growing in the St. Lawrence River. It's a small shark, don't worry. You can go and swim in the St. Lawrence. But you know, the genome has not been completely sequenced or annotated, so it's more demanding. But most of the time, we succeed in having a good glycogen signature. Uh, Another thing that we do a lot, we've been doing that for years, is microbiome analysis. Um, I know, I mean, we can help you there without any problem. In this cartoon that you see, the sample collection is feces from humans. We do it from any animals, actually. We do it more often in terms of animals. But we have specialized over the year in the circulating microbiome or the blood microbiome, which is way more difficult to analyze than feces analysis. There we can help, whether it's human or animal, any kind of animals. We've done the microbiome analysis. So we can help you with DNA extraction, sampling, the sequencing in all the analysis. The microbiome analysis is pretty straightforward, but sometimes you have to be careful. Most of the times, the analysis, as you see in the middle over here, is targeted the V3, V4 region, bacterial gene of the 16S RNA gene, and that works well. But we do it, we've started to do more larger regions like V1, V9, which allows you not only to identify bacteria at the genus level, but at the species level because you have a longer region. And we also have done microbiome. We can provide help into microbiome analysis using shotgun sequencing, so a metagenomic bacterial sequencing of the blood sample or any blood samples or even plants. We've tried and it works really well with plants. And I give you, for those of you who want you know, a more detail of what we've been doing in microbiome analysis. I've just put our analysis here. We've done it into different kind of fish. We've done it in Antarctic seabirds recently. We just published that recently. We do it in blue mussels, the cod, halibut populations, the trout, salmon also. And this is always tricky. So if you get into that for people working into the environment, we certainly can provide a lot of help for this because um, this is years and years of experience, and that helps a lot to accelerate your research. Um, some things that we can help, and we help people for that, is the nanopore sequencing technology. Um, most of you who have done DNA sequencing or RNA sequencing analysis and sequencing analysis have been using probably Illumina, but now with the emergence of long read sequencing like PacBio or Nanopore, we've done that. This is an example where we have our own DNA sequencers because they're so small and so inexpensive, and these are very useful. This paper is an example of the how we can help. If any of you guys want to go and do some DNA sequencing in the field, we certainly can help with the pictures that you see there is a research project that we had with the French government in Antarctica, in a research base in Antarctica in Kerguelen, Ireland. And our project, our mission was really to demonstrate if we could do DNA sequencing on site. And on site means no internet, no electricity, no heating, nothing. And what you see at the bottom right is our wet lab into one of these abandoned shed that was abandoned for 50 years. So we've set up a lab, and you see the DNA sequencing, the DNA sequencer over here. And so we were picking up samples of blue mussels at site 1, 2, 3 and the Armour Lake, the Lake Armour. And we were actually picking up the samples, doing DNA extraction, library amplification, and DNA sequencing into the nanopore sequencing. Just to let you, this is so efficient. I mean, we sequence the equivalent of the human genome in 3 or 4 days using that very small sequencer over here. So it's very powerful method. Okay, and okay, bioinformatic analysis. I talked to you about it. One thing, a lot of people can do microbiome test about it, but I talked about it. One thing that we like to do is the virome, looking at viruses, and these are the data that we have got from our nanopore sequencing into Antarctica. But you can do it everywhere. But gives you an idea here. But if you get a very deep sequencing with the nanopore technology and long reads, we get reads up to 100 kb sometime. You can identify all the viruses that you get into your samples. The samples being here DNA that was extracted from blue mussels. And this is something that we like to do because viruses has a lot of progress on that. Do we have questions or just a mic, an open mic? Okay. And because time is flying, this is another example of a virome analysis from circulating cell-free DNA. So this is viruses that you find into a blood DNA. This is very an emerging concept. You take a piece of blood and you look at all the viral DNA that you can get, and that now it's being used in clinic to determine, the presence of an infection, an ongoing infection. Um, I won't talk about production of recombinant proteins. We can do some of it. Just contact us, and then we'll see if we can help you. I just want to spend the last remaining time on the generation and characterization of nanobodies. I think this is something that is getting more and more popular. A member of Glyconet recently called me last last week because he wanted to develop nanobodies. So I helped him out, and we are offering our expertise for the development of his nanobodies. So what are nanobodies? I'm an immunologist. For me, when I've heard about nanobodies, I thought this is a fantastic discovery. What you see on the left is your kind of antibodies that you are used to and that you have in your own blood. But about 20, 30 years ago, it was discovered that the nanobodies, the antibodies in the camels are very much smaller. And actually, the recognition site, the site of the antibodies that binds to the antigens, is only involves only one chain, whereas you need two chains for the human antibodies. The consequence of this discovery is that you can generate very small nanobodies based on this discovery. So the advantage of this is that they are very small. So for us working on cancer, it's uh, a great advantage because for solid tumors, these large antibodies have a hard time to penetrate into the solid tumors, whereas the nanobodies can penetrate the solid tumors more deeply. And if you conjugate them to a toxic agent, a radioisotope, then you can have a better targeting of your tumor. They are very simple to manipulate. It's a single domain structure, so it had, and they were designed. They have high solubility, very stable. Um, it doesn't cost anything to produce. I mean, this is compared to antibodies. It's remarkable in terms of cost of production. You can produce hundreds of milligrams for very cheap. Very easy to for engineering, better tissue penetration. I already mentioned it. And in terms of, I'm just confirming. I don't know a decline. I have to do it, Elisa. Confirm decline. I'll just confirm it. So I don't want to get. And what is nice about, I don't want to talk into details, but the nanobodies recognize the antigens differently than the traditional antibodies. And they have access to hidden epitopes. And this is a big advantage because there are some antigens that are very hard it's very hard to develop antibodies against these antigens. And the way the nanobodies recognize the antigens is it makes it way easier to do it. Uh, it can be done rapidly. Uh, so I'll just finish with the nanobodies. Just to give you an idea what we can do with nanobodies, they're very small, easy to produce. You can do bispecific, you can do mini bodies, depending on, and you can do bispecific antibodies on this. Um, so a great example, if anybody was working on brain right now, there was a great paper recently in Nature where they developed these bispecific nanobodies, and they showed they can access to the brain through the brain barrier, and this is a great advancement, and that will certainly open a lot of possibilities for targeting antigens into the brain. Another a big concept that or discovery that was recent is we can deliver antibodies for immunotherapy or using subcutaneous injection instead of intravenous injection. And even there are some indications that we can deliver these nanobodies or these antibodies orally. Just you would take a pill and could get a treatment with the antibody, and that would certainly reduce a lot of the cost and the logistics associated with this treatment. So we can do, from the conception, we can have from the conception to the purification and also for the characterization of the antibody. If you want more about that, we've just published a paper where we describe the whole process from the discovery to the application of a specific nanobodies here. So this is it. I mean, We're not, as you've seen, we don't have a lot of big equipment, but we have a lot of expertise. We do a lot. Most of our equipment uh, is based in other core facilities, like Genome Quebec, Eve Technologies here in Calgary, Hybridgenics, Cynobiologicals, of course, the Digital Research Alliance of Canada. We work a lot with that, especially for the bioinformatic analysis, because these core facilities are just fantastic. And give you a few examples of collaborations and associations or customers we had over the recent years. It goes from academic to governmental organization in Quebec, Canada, and also in France. And so, yeah, this is it. I hope you had a good idea of what we can do. As the image shows here, this is not my research assistant. This is AI created. We're waiting for your calls. Do not hesitate. Contact me by email, and we'll see if we can help you. Any questions now?
Thank you, Eve, for presenting. Sorry about the interruption from the random person. Yes, and go ahead. Someone has a question. I think that's Warren, but accidentally me.
Yes, it was. It was me. Eve, thank you very much for that lovely presentation. Can you tell us approximately how long does it take from, starting on making a nanobody to sort of ending up with a usable version?
I would say now with the synthetic library, when we start, I would say 3 months.
Okay.
That would be the fastest. So I would say anytime between 3 and 6 months. It depends of your protein, but in some cases we had a turnover, and because we go to Hybridgenics, we provide the proteins, they do the screening. That doesn't take long, screening. It's a phage library. They subclone it into the vector, expression vector, bacterial expression vectors. They send you the vectors, everything is sequenced, and then you produce it in your own lab. They can produce it for you if you want, but we produce it in our lab. So to produce it, it takes a few days, and you can have like, hundreds of milligrams of these proteins. We like to use hybridgenics for many reasons. They were among the first to generate a synthetic library. Uh, they were very smart in terms because their 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 uh, their uh, um, their framework has been optimized for stability, so resistant to pH,
Okay.
stability, thermal stability, immunogenicity, so they're not immunogens, and they're very stable inside the cells. You can take these nanobodies and target intracellular proteins, using
Okay.
PROTACs, for instance.
Yeah.
And they will generate all the vectors you want. You can use other companies to do that, but they'll do it because they already have the clones. And generating PROTACs is like 100 or $200 for the plasmids, was really cheap. So,
Okay. All right. Thanks very much.
welcome.
Lisa?
Hi, Lisa.
You've been talking a lot about the nanobodies recognizing proteins. Has anyone tried getting them to recognize glycans, and how's that going?
I don't know, the glycans, the problem is that they're poorly immunogenic. I don't, but if I've never tried myself, I haven't seen any doing that, but I'm not aware of the literature. I'm not up to date on the literature, but I would bet yes, because of the way they recognize. They have, for those who are an immunologist, we have the recognition of an antigen is made by the combination of CDR1, CDR2, CDR3, which are at the end of the antibodies. And what is great about the nanobodies is that the CDR3 is a long range, has a long amino acid sequence, whereas the traditional antibodies are shorter. So they have access to to places or immunogenic places where the traditional antibodies have failed. So, but maybe someone has the answer in the audience, but yeah, it's certainly worth trying. I would be confident.
Dawn, go ahead.
Oh, a second here. Okay. You said you had quite an extensive selection of cell lines. Like, is there a list on your website, or do you just have to inquire, or?
Inquire. We have a lot of cell lines. We haven't put it on the list. We have too much work, but we know what we have. We have we have we have an Excel sheet that describes all the cell lines we've got over the years from, collaborators or generated by ourselves. But yeah, just send us an email and then we'll see if we have it.
Okay, thank you.
Actually, Elisa, the next step would probably to set up a website. Every time, I say, oh, I have to put it on the web. I have to set up a website. I promise I'll do it one of these days.
We have the services, like an integrated services page. So if you have materials that you want to put on the website, we can discuss.
Oh, okay. Perfect. Thanks. So anyways, so if anybody has any question, just email me and then we'll try to help you as best as we can.
Okay, thank you everyone for joining, and thank you, Eve, for presenting.
Good luck with the editing.
Yes, have a good rest of your day, everybody.
Thank you, everybody. Bye.